अस्सलाम वालेकुम गाइस, आप देख रहे हैं मेरा यूट्यूब चैनल। तो आज हम टॉपिक डिस्कस करने वाले हैं वो है ड्रग्स ट्रीटमेंट एंड जीन थेरेपी। इफ अ पर्सन becomes sick with disease, it is subjected to the action of antibiotics which can kill bacteria. The antibiotics are however useful in bacterial disease, and they and that only when bacteria have not developed resistance to antibiotics. In cancer, radiotherapy and chemotherapy are also be used. In radiotherapy, the cancer part is exposed to short wave radiation from the radioactive material repeatedly at regular intervals. In Pakistan, there are several centers which are carrying out radiotherapy to control cancer. Chemotherapy consists of administering certain anti-cancer chemicals to the patient at regular intervals. These chemicals may kill both cancer and normal cells. Recently, a new technique has been developed to repair affected genes. This consists of isolating the normal gene and injecting it into the host through bone marrow cell. This is called gene therapy. Combating disease utilizing all methods has and when required are ensure a participation of community in this program is known as integrating disease management. This required awareness of the community about the severity of the problem it causes and its remedies. This is a very effective program for elimination and control of dangerous disease from the human society. Besides its contribution of food protection and health of women, biology has discovered a number of means and developed techniques for the welfare of mankind, as for example cloning, protection and conservation of environmental etc. Cloning is a technique for achieving energetic aims. A clone is defined as a cell or individual and all it asexually produce offspring. All members of a clone are genetically identical except when a mutation occurs. Generally, no normal animal reproduce naturally by cloning. Several insects and many plants do in some circumstances, whereas few do so regularly. In 1997, scientists in Scotland succeeded in cloning a sheep. Other mammal species, maize and cows, have since been cloned. In the procedure, the nucleus from a fertilized egg is removed and a nucleus from a cell of fully developed individual is injected in its place. The alternative zygote is then implanted in a suitable womb where it completes its development. The new individual formed in this way, it is a genetically identical clone of the individual whose nucleus was used. Thus, cloning could make multiple copies of a desired genotype. Another type of cloning is the division of a single egg. or early embryo into one or more separate embryos. This is the same process that normally creates identical twins. Offspring from this type of cloning are genetically identical but carry chromosomes from each of the two parents. This type of cloning has already been used to produce genetically identical cattle and other farm animals. Man is likely to adopt cloning technique for commercial reproduction of valuable animals of known pedigree such as horse etc. At some places, scientists are making attempts to clone human embryo which they believe can survive as transplant donor. There is a lot of controversy on this issue as to whether human cloning should be attempted or not. तो ये था आज का हमारा टॉपिक ड्रग ट्रीटमेंट एंड जीन थेरेपी। अगर वीडियो अच्छी लगी तो वीडियो को कर लें लाइक, चैनल को करें सब्सक्राइब। मिलते हैं नेक्स्ट वीडियो में। अल्लाह।